So this is the last path of photosynthesis. I'll look at the cyclic electron flow. Now, don't forget we look at the linear electron flow. Our electron flows from photosystem 2 to photosystem 1 in a linear fashion, right? So that's the major thing, actually. But I just want to touch base on this. So in cyclic electron flow, the electron that is photoexcited, right, is going to, there's electron that is photoexcited electron back from, we go back from ferrodizine to cytochrome complex. So it comes from here, go back to ferrodizine to cytochrome complex instead of it going to NADP plus to become to reduce NADP plus. Electrons are then passed through P700 and then chlorophyll in the PS1. So it goes this way in a cyclic pattern, then it goes to P700 in PS1 through PC here, which is plastocyanine molecule. So the cyclic electron form uses PS1 only, right? Instead of using PS2. So it just goes that way. And it does not include this. It does not include this. So it's more like the photon hit on the antenna complexes. Then it goes to the reaction center, gets it excited. Then the electron goes round again and come back to replace the electron. So the major thing is that in the case of that, ATP is produced, but it will not produce any DPH. All right. So cyclic electron flow uses only photosystem 1. It doesn't use photosystem 2. It produces ATP, but no NADPH or oxygen results. Because if you think about it, there will be no need for oxygen because the purpose for oxygen is because water molecule, the purpose for water molecule splitting, right, is because it has to replace the electron that was here because it's a linear pattern. All right, so the electron lost in P680 has to be replaced by splitting water molecule, and the cause of that, it will eventually release oxygen as well. But in this case, oxygen, there's no need to replace any electron anyway, because the electron that was removed still came back and replaced itself here. All right, so that's why it's called cyclic electron flow. All right, so let's look at, does that mean that cyclic electron flow can occur without? Yes, that's the meaning. That's the meaning. You see here, first is it does not require fuel system 2, and it's also not going to produce any DPH because it does not require the splitting of water molecule to produce that. All right. So several groups of photosynthetic bacteria, cyanobacteria is one of them. They have only single photosystem related to either PS2 or PS1. For this organism, cyclic electron flow is the only means of generating ATP during photosynthesis. Because they only have photosystem 1, they can only undergo cyclic electron flow. They cannot do the linear because the linear require two photosystems. It requires both photosystem 2 and photosystem 1. So some of them only have one photosystem. So photosynthesis may have first evolved in the ancestors of these bacteria, for example, like photocyanobacteria particularly, in a form similar to cyclic electron flow. Cyclic electron flow is probably in path and evolutionary leftover in organisms with both photosystems. So organisms that have both photosystems will of course undergo linear, but organisms that have one will undergo cyclic. So cyclic electron flow may have photoprotective capability. plants that do not have it grow well in low light. So plants that do not have it grow well in low light, but plants that have it that but cannot grow well in intense light. So we say they have photoprotective capability, meaning that they have capability to protect, to be shielded from sunlight. So they grow well when there is low light. Whenever there's low light, they are able to grow well because they don't have capacity to retain too much light. And you should think about it, a system that has two photosystems and the light can be captured by two photosystem and a system that have just one photosystem. The one that have two photosystem have capacity to embrace enough sunlight, but the one that have just one photosystem have limited uh, anatomy to absorb sunlight as well. All right, that's major thing I need you to know about it. So a comparison of chemiosmosis in chloroplast and that of mitochondria. So it's more like you're looking at cellular respiration and photosynthesis now. Chloroplasts and mitochondria both generate ATP by chemiosmosis, which we just saw. And I told you chemiosmosis is what happens in the electron transport chain in cellular respiration because it does not involve, it does not, I mean, it's not substrate level osmosis, right? Electron transport chain pump proton ion across a membrane as electrons are passed through carriers with progressively higher electron affinity. The ATP synthase is going to couple division of hydrogen ion down their gradient to phosphorylation. I mean, to phosphorylation of ADP and ATP. This is what I explained to you in cellular respiration. If you have forgotten that, you want to go and review it again. Now, some of the electron carriers, including ion containing proteins called cytochromes, that's part of the electron transport chain, right? Are very similar in mitochondria and also in chloroplasts. They are in both organelles. The ATP synthase complexes are also very similar. The ATP synthesis, they are also very similar. Phosphorylation differs from oxidative phosphorylation in few key. In chloroplasts, high energy electrons drop down the transport chain. I'll come back. I didn't get the question very well before it goes off my screen. And because I'm sharing my screen, I'm not able to see it now. So I'll come back to it. So in chloroplasts, high energy electrons drop down the transport chain from water, while in mitochondria, they are extracted from organic molecules. You remember we've talked about that in mitochondria, you get it from glucose, right? But here it's from water. Mitochondria transfer chemical energy from food to ATP. Chloroplasts transform light energy into the chemical energy of ATP. This is making, the other is breaking down. All right. Although the spatial organization of chemiosmosis differs slightly, there are similarities. In mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis by powering the ATP synthase as it diffuses back to the mitochondrial matrix. But in chloroplasts, proton are pumped into the thylakoid space and drive ATP synthase on the stroma side of the membrane as it diffuses back to the stroma. So they're about the same orientation. Let's go. All right. Now, let's go ahead and talk about the dark reaction, which is what I want to talk about in very few minutes. We've looked at what is the major part of photosynthesis anyway. The dark reaction or the light independent reaction or Calvin cycle is just those three stages there: carbon fixation, reduction, and then regeneration of the substance. So both ATP and NADPH that we produce in light reaction are going to be the raw material that will be needed here. Those are the guys that will feed in the Calvin cycle to cause generation of sugar. So both ATP and NADPH are produced on the stroma of the thylakoid membrane, making them available for sugar synthesis in the carving cycle. All right. Okay. Now, this is what we are talking about. You see NADP plus, NADP, an H plus becoming NADPH. And this is ATP here. So this is light dependent reaction. You see the photosystem 2, photosystem 1, how the linear process goes on without happening in a circular manner. And then NADPH produced here and ADP produced here comes straight into the carving cycle, which happen in the stroma. All right. Now, let's go. Now, the carving cycle, like the citric acid cycle in photosynthesis, it always regenerates its starting material. If you remember when I taught you, cellular respiration rather. When I taught you cellular respiration and I told you about the citric acid cycle there, I told you something. I told you that oxaloacetate, which is a 4-carbon compound, was waiting in the citric acid cycle, waiting for a 2-carbon compound, which is the reason pyruvate has to be reduced to acetylcholine. Do you remember? Now, and after the whole process, the the oxaloacetate has to be produced. It has to be restored, waiting for another acetylcholine. The same thing here, you see, the carving cycle, like citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. The carving cycle is anabolic because it's building up. Cellular respiration is catabolic because it's breaking down. It builds sugar from smaller molecules by using ATP and reducing power of electrons carried by NADPH. So it's going to use both ATP and also the electromotive power, electron power in the electrons, in the NADPH rather. So the carbon is going to enter the carving cycle, but they are going to enter as CO2. Don't forget, glucose, each carbon in glucose leave cellular respiration as CO2. In this case, each carbon that comes into the system comes in as CO2, right? And leaves as a sugar molecule named G3P. Now, what is actually produced in photosynthesis is a 3-carbon compound, which is actually a sugar molecule as well. It is not carbohydrate, it is not glucose that is produced directly. All right, glucose is a polymer of what is produced. Now, for net synthesis of 1G3P, which is glyceraldehyde 3-phosphate, 3 cycle must take place. Now, the cycle must take place 3 times. What does that mean? If 1 carbon dioxide comes in, we've only had a carbon. Another carbon dioxide have to come in so that we have 2nd carbon. Another carbon dioxide have to come in so that we have the 3rd carbon. So for a glucose molecule to be produced in essence, there has to be 6 carbon dioxide has to be imputed into the carving cycle. That's what it means. The cycle have to run 6 times. So to get G3P, which is a 3-carbon compound, there has to be 3 times of the cycle in code, right? So for net synthesis of 1G3P, which is a 3-carbon compound, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide, 1 each turn. The carving cycle has 3 phases. It has a carbon fixation, it has the reduction phase, and the regeneration of the CO2 acceptor phase. Just the same way that cellular respiration have it. Let's go. So we are going to look at the carbon fixation. Once we look at the 3 stages, that's it. So the carbon fixation phase is this. Let me get my pointer. Now, in the carbon fixation phase, it is simple as thinking about it like that we are fixing carbon, as though we are storing carbon. We want to fix carbon to make glucose. So the binding of CO2 to a 5-carbon compound, which is what is located in the Calvin cycle, waiting for a carbon to come. So it's a 5-carbon compound. We call it RUBP. Okay, I'll talk about that. Now, and it's catalyzed by RUGP carboxylase oxygenase or Rubisco. So it's either catalyzed by Rubisco or this name. Now, this is what you have here. This is RUBP here. It's a 5-carbon compound waiting for carbon to come each time. So the 6-carbon intermediate is immediately split into 2. So this is once a carbon comes in and add up to this guy, it's going to form a 6-carbon compound. This is a 6-carbon compound that is formed. You see that? Immediately the 6-carbon compound will be split into 2. So it will be split into 2. Now that's what you see. So the 6-carbon compound intermediate is immediately split into 2 molecules of 3 phosphoglycerate. All right. So 2 of these will be produced straight from these 3 compounds, this 6-carbon compound straight away. All right. So again, carbon comes in, join with the 5-carbon compound, form this 6-carbon compound. Immediately, the 6-carbon compound is going to split into 2 of phosphoglycerate. Now that's the first thing that happened. That's phase 1. Phase 2, which is the reduction stage. So all that we call carbon fixation is simply that the carbon comes in, joined to the 6-carbon compound, to the 5-carbon compound, split into, form a 6-carbon compound, then split into 2 phosphoglycerate. That is what we call carbon fixation. Now, reduction phase of it. The major thing about the reduction phase, so each molecule of 3 phosphoglycerate is altered through phosphorylation by 6 ATP and reduction by 6 NADH. So you see, let me do my annotation. So you see, this is 6 NADPH invested there. This is 6 ATP invested there. So 6 ATP will phosphorylate the 3 phosphoglycerate. So you see that each molecule of 3 phosphoglycerate is altered through phosphorylation by 6 ATP and reduction by NADPH. What does that mean? It means that ATP is going to donate phosphate to the 3 phosphoglycerates. Number 2, NADPH is going to reduce it by contributing electrons to it. When they receive electrons, they become reduced. So when that happens, they are going to form G3P. And G3P is, okay, I'll talk about that later. So they form G3P, and that is actually what we call reduction phase. So for every 3 CO2 molecules that enter the cycle, 6 molecules of G3P are formed. Now, only one of these can be counted as a net gain of carbon dioxide, of carbohydrates. So there is one that will be noted as this is the product, G3P. Let's go. Now the third phase there, which is the regeneration of RUBP. So the remaining 5 molecules of G3P are rearranged in a complex. They are rearranged in a complex of series of reaction yielding 3 molecules of RUBP, and 3 additional molecules of ATP are used to facilitate the process. So all that matter here is that at the end of the process, after carbon fixation, after reduction, you have to restore RUBP and be produced back. Now, in the course of production of RUBP, major thing you need to know is that 3 ATP will be invested in that process. So that means in all, 9 molecules of ATP are invested in production of glucose molecule, and 6 NADPH are made. So in the old sense, at the end of the process, ribose biphosphate, which is the RUBP, which is ribose biphosphate, will be produced back in the cycle, and that is what is important about the process. So the summary of Calvin cycle is that there is a 5-carbon compound, which is ribose biphosphate, RUBP, waiting inside the cycle for carbon to come. Carbon is going to come in form of CO2. It comes in, deposits this carbon, and then a 6-carbon compound is going to be formed. Ribisco is the enzyme that powers that reaction. Once a 6-carbon compound is formed, it immediately split into 2. It's split into 3 phosphoglycerate. The phosphoglycerate is going to be phosphorylated by 6 molecules of ATP, and is going to be reduced by 6 molecules of NADPH. At the end of the whole process, a G3P will be formed. Now, the phosphorylation is what led to 1,3-biphosphate to be formed, because it was just one phosphate here before. And then the reduction led to production of G3P eventually. And that's pretty much what happened on the reduction phase. Then the regeneration phase simply requires that 3 ATP will be invested to make the RUBP, ribose bisulfate, back into the system, so that another carbon can be fixed again. And it goes straight in that pattern. Let me see. Okay, I thought I was going to play. All right, so assessment of the whole cycle. For the net synthesis of one G3P molecule, the Calvin cycle consumes 9 molecules of ATP. Like I told you, it's going to consume 6 molecules of NADPH. The light reaction generates ATP and NADPH. G3P is the starting molecule for the metabolic pathway that will produce organic molecule that we call glucose, sucrose, galactose. So what is actually produced? It's G3P. Then G3P is not going to be the precursor that will lead to the formation of others. And G3P is glyceride aldehyde 3-phosphate, G3P. All right. Alternative mechanisms of carbon fixation have evolved in hot, arid climate, dehydration. I think I'll just leave you to read that. There's nothing much about that I want to teach again. Yeah, I'm going to stop there.